It's just the one here, right?
Yeah. Unless we should take a double.
What's that?
Scientists, do you think we should take another, or do you think one is sufficient?
Was Steve saying we should take two? One for the top few inches for you and USGS, and then the other for that group that's doing the microbes?
Yeah, I think that sounds good.
You guys mind if I move just a bit?
Yeah, it should be fine if you move just a bit.
Roger.
And the push core we just took was sample 062.
Stirred up here.
Roger.
Thanks.
And we can go ahead and treat this as a duplicate sample, so we don't have to give it a new sample number since it's in the same spot.
Okay, so 6-2 will be push core number 1 and what next one?
Yeah.
Just gonna move forward a little bit and change my heading so I can swing around without. So one more core for sure, right?
Yes, please.
And turn off the porch light. And show me the azimuth on bubble. Thanks. Are you guys happy I haven't disturbed that, or do you want to move forward a bit?
No, I think a sediment core anywhere is fine.
Yeah, I think that looks just fine.
There we go.
Well, that's a nice core.
Nice.
It's
Oh, took too long to come out.
Yeah, the other one stayed in there pretty nicely.
What's that?
That first core wasn't budging.
This one slid out, so I'll have to try again. See if I can come around a little faster this time. So you leave that bubble cam on the azimuth there for me. Oh, come on.
Nice.
Okay. Put our suction sampler away. We're done with that, right? You can
Yeah.
Yep.
turn the porch light back on.
Our next objectives will be to get 100 meters between us and the last transect and start another.
Sure thing. We'll be set up pretty good here.
We can come out of salvo too.
Yep.
You want to pull the porch in just a bit for us?
Coming in.
That's good. Yeah, come all the way in, I think.
All the way in?
Yeah.
Roger.
Really? Oh, come on, Dan. Switch that other camera to the rail cam for me. Be great.
We're actually set up in position here. I think we'll move up this slope at 030 until we get a bit flatter.
Roger.
And start the ship if you're happy with that.
Yeah.
Bridge nav. Step 100 meters, bearing 030. Thank you.
was one muddy ROV.
He's shedding little mud into our view for a little while so all the stickiness comes off.
Can come down a little bit, Jake.
Going down.
Yeah, it should come off when we start going forward there. Wide to left a little and get rid of some of it. Transecting or just cruising?
Cruise until we get
once we've
100
got-
away, and
Oh,
then
roger, yeah.
Yeah.
Then a
transect after. Sorry, what was that?
Oh, you were just saying what I was about to say. Perfect.
Okay.
About how far away are we from point AB?
Point AB is 460 meters away.
Okay.
We can pick up the speed a little if you want.
Yeah, that'd be great.
Sure. Bridge nav. Increase to 0.3. Thank you.
Yeah, in the meantime, we'll be watching out for siphonophores and Argus.
Favorite pastime.
Back into the mud.
Well, we started around 800 meters and we've now climbed over 100, about 150 meters. And point AB is going to be right around 500.
Okay. Doesn't it flatten out and then we spend some time on the spot for part of it?
Mm-hmm. Yeah.
Nice flatfish coming and going.
Yet another species of sea star that we've come across quite a few so far.
Nice little myctopids making appearances. Little pout down at the bottom. And tiny little thorny heads.
Bob texted me that the assassin bug is capable of inflicting a very nasty bite.
The assassin bug?
Yeah, the bugs that are all over the boat.
I
Oh
haven't,
no,
caught-
really?
I haven't seen him.
I caught one right when we were doing the daisy chain.
Ah,
Really, that's interesting. I don't know if I've seen assassin bugs here while out on the vessels.
Well, that's what we think it is. I'm no entomologist, but my oldest is right
Saw a
into
weird
it.
looking bug when I was collecting the Niskins earlier. I'll Google it and see if it's the same bug. What I saw.
I've got one captured in a electrical tape container on the workbench, and
Yeah. Yeah,
They-
something like that.
Are they flying insects?
I'm gonna
It
go,
almost looked like a stink bug,
I'm gonna go look at it.
Oh,
but it looked a little bit
ew.
like an assassin. The pictures here. I saw a few, one in the
They're
wet
not-
lab, one in when I was getting the Niskins.
When and how did they get on the Nautilus?
I don't know.
Well,
That
they
is
are
so
beetles.
strange.
They can fly.
I know, but how far? We've never seen them while out on our vessel. We get a lot of kelp flies.
We got a little snailfish.
this,
Yeah, that was a pink
Oh,
snailfish.
cute. Cute.
I've been dragging bugs home from all over the planet for
Don't do
the
that.
last 5 years. So my kids are in entomology and 4-H. Our 4-H leader is a retired entomologist from OSU, so
Oh, cool.
she's really good. Really good with the kids. One of her kids always takes a blue ribbon every year. So, the last couple
That's
of years, it's
impressive.
been my oldest.
It's one of my first science jobs. In college, I worked in USDA quarantine beneficial insects research laboratory, and I basically was in charge of a colony of 1,000 Asian longhorn beetles.
Oh, wow.
That was terrifying.
Yeah, they're big
Yeah, they make little grunting sounds and
when they do.
pinch you.
Oh,
Why did
what
I
do
do
they
that?
eat?
Maple
Huh.
trees. That's what they eat.
Oh, I thought you said people.
Well, that's where they burrow for their laying eggs or whatever.
Have they released those engineered mosquitoes in Key West yet?
They did?
How's it going?
I haven't heard what the outcome was. I just heard they were pressing on with the release.
Mm-hmm.
That's the last I read about it.
Are these the ones that can't get Zika?
Sorry, say again?
Are those the mosquitoes that can't carry Zika?
No, I think
Or just the ones that are
they're engineered
sterile?
to, yeah, not reproduce. I think it's the, I forget, one of the sexes is engineered to not be able to reproduce. Kind of scary
Hmm.
in some aspects of that.
Bridge nav. Add one more step, 100 meters, 030.3. Thank you.
Given what we've seen for the last hour, I don't think Matt is off the hook for a mud dive yet.
No, I was just thinking that.
We're reaching a less steep slope, and we've just finished our 100 meters if we want to start a transect here, knowing that it'll be not as steep.
Yeah, that's fine. Yep. And the speed we're going is good too,
Okay, sounds good.
especially since we're not seeing a whole lot.
All right, we'll start it there. Taylor in.
Yeah, we're at 0.3 now. I think we can, logistical wise up here, we could hit 0.5.
Sure.
If you guys are all right with that.
Mm-hmm. That sounds
Yep,
good.
that'll be fine.
Okay, I'll increase to 0.5.
Roger.
Bridge nav. Increase speed to 0.5. Thank you.
If Tom gets got some hard substrate on his first transect, he can get soft substrate on the second.
So if you look at high pack, which is up on channel 3, you can see these red rings at the end of it is 200 meters. Those are 50-meter rings. And I figure when we get up here, we could either lateral over in this less steep slope area and then go directly up to AB, or we could choose a path however you want to get up there at an angle or straight on, and
Yeah, I'm open.
Sure.
Seeing what habitat we encountered.
Yeah, just depending on what we're seeing, it might be nice to lateral over and catch that kind of steeper looking ridge line.
Yeah, I agree. Kind of a ridge looking thing might be interesting. Or...
Yeah, I like that. What kind of fish is this? Is this hake, sablefish?
Just saw the tail. It looked like a sablefish from the tail.
Okay.
Hake will have a long fin that goes all the way down their dorsal side, and then sablefish will have multiple fins.
First sablefish sighting of this type.
Oh,
a bunch of fragile pink urchins.
They're so small.
Yeah, nice pile of them.
Cluster of them. Look at that.
See, we're not on transect, right?
We are.
We are on transect.
Sorry.
That's all right.
Lost the plot there for a minute.
That's okay. I think we needed to go over to starboard anyway to stay out. Interesting patch, though. I wonder if there was something there at some point.
Well, that was a nice passing. Give us something to look at for a few seconds.
I apparently just woke up my poor dog, who's now like, why are we up this early?
What kind of dog?
He is a chocolate lab.
Ah,
Oh,
happy dog.
Yep.
What's his name, Meredith? I love his name.
Satchmo.
Satchmo. That's so cute. How did you come to name him Satchmo?
My husband Russell loves that name. And so when we first got married, we made a pact that our first serious pet or child would be named Satchmo.
Pet and or child.
So pet it was.
wasn't that Louis Armstrong's nickname? Am I making that up?
Yep, that's Louis Armstrong's
Louis
nickname.
Armstrong, yeah.
When we got off watch last night, I saw my friend Sarah, who's also a video engineer on Nautilus. She had texted me. She said, "Is there a dog in the control room?" 'Cause I think she was
Sarah
watching it.
Matisic or Matasic?
Yeah, Sarah Matzik.
Nice.
Probably this guy barking in the background.
Yeah. I think the first time he barked yesterday, we all reacted, and then I think he barked later, and we just all ignored it. So that's probably what she heard.
That's
And
right.
then later she was like, "Oh yeah, I forgot that there's scientists on shore." She hasn't been on this year.
Yeah.
ship dog would be cool
Bridge
though.
nav?
Yep.
Right? Like a Nautilus mascot? Non-sea themed?
Add one more step, 100 meters. Thank you.
Seeing a little bit of seagrass.
Today it appears to
That's
be
fun.
not.
You're at it's a non-stop.
Sponge.
I don't understand that.
I don't either. Has to do with angle, it must.
Yeah. Today it's a non.
Well, some watch based on math prediction is going to get some good hard bottom.
It just may not be ours.
It just may not be. Well, maybe on our second watch today.
Fingers crossed. I think I might have backscatter for this, probably from the Nautilus. Might pull it up and see.
Yeah, let me know. I didn't have I didn't double check to see if we had backscatter in this area. I looked on the last dive, but the last dive we did not have Nautilus coverage there.
Can bring your head to the left a bit if you want.
I can.
We do towards AB, so I'm hoping it looks like we have some stronger returns towards AB where we'll find some more hard habitat. So yeah, AB through 85 has backscatter.
So it's just this lower part that's potentially muddy or actually muddy.
Hopefully. I'm not super familiar with backscatter, and I also wonder if there's like sediment on top of hard bottom. Would we know? It would just look like sediment, you know?
That can also lead to a high backscatter return. You're right.
Yeah. Yeah, I don't know how deep the sediment has to be before it's a soft return versus a hard return.
It's a $1 million question. We do have data here, Nautilus data, so I can get some.
Yeah, if you're interested.
I can get that backscatter up. Can I do it right here? Let me think about this. I don't have NB Proc 2 button.
For anyone who's joining us from home in the middle of the night or during the morning hours, if you're over in Europe, the backscatter we're discussing is a type of sonar mapping that tells you how hard the substrate on the bottom is. So when we're looking for corals and sponges, we're typically looking for hard bottom habitat. Right now we're out in the soft mud and working our way through a quantitative transect, which is a method we use for comparing sites coast-wide. All of the invertebrates and fish that are being seen during these transects get counted, and then because we use the same methods up and down the coast, we're able to compare between sites.
How often do PIs ask you guys to do transects? Because often I feel like the oh gosh, it is 3 in the morning. Primary mode of transit is really like an exploratory nature versus directed headings and speeds.
I think it just depends on the objectives. If they want, if they have someone to review the video and they want a constant speed and bearing, some groups do a few transects, some do 0. This seems to be some of the most, I'd say, transects on the dives that we've been doing for a couple of the national marine sanctuaries along the west coast.
Yeah, this has been a coordinated effort this year.
Yeah.
To be transect heavy.
Bridge nav. One more step, 50 meters, 030. Thank you.
But we do tend to always have waypoints and points of interest. Right, Rennie?
Yeah, we do some, it depends on the group, but they'll have dive planning kind of done in advance, or sometimes if it's exploratory mode and it's not necessarily for a specific group, then sometimes we'll do a lot of mapping and dive planning on the fly right before the dives. Very exciting. Mapping something that hasn't been mapped, turning it over, picking a route, and exploring it right there.
Mm-hmm.
Yeah, you're doing that to some extent for Santa Lucia, right? Which is going to come up over the weekend, potentially?
That's correct. Yeah, there's been a fair bit of mapping actually there by a few different groups of Falcor, Nautilus, Rainier, and kind of amalgamated them all together.
When different groups map, is all the data packaged in the same format so you can combine them easily, or do you have to do it kind of?
Depends. There is the raw files, the point clouds, the XYZ data that we're able to. We can ingest that and kind of combine it with ours, or oftentimes they will have made rasters, standardized grids of varying resolution. And something we run into, especially with earlier operations, is backscatter was not collected or recorded, and so that's just the intensity of the return of the signal. So, and even more recently than that was water column is pretty dense data, so it was kind of prohibitive to record it in earlier operations, but now becoming more and more standard. So water column is where we can find a lot of cool information about biological layers, but something we do more often on the West Coast, especially, is methane seeps. That's the edge there of the 200 meters. We're all done with this transect.
Okay,
Awesome.
nice.
Bridge nav.
We got that little black-tailed snailfish right in time.
Yeah.
Hold position. Just stop this here, and then we will lateral over H1828. Okay, let's see if we can lateral bearing 290. Or if it might not be, you might be able to drive straight on 290 because it's pretty flat.
That DVL could use some love.
Roger.
Two nine
290.
290.
Bridge nav. $100 meters, 290. Thank you.
Sea cucumber, maybe?
Looks like it.
I think that looks like the sea cucumber. Yep.
Looked odd. Wasn't sure.
It's kind of flattened on one end.
Yeah. Got half-empty toothpaste.
Good we sampled those sea pens, because right after we sampled them, we got out into this flat mud where there had been far, far fewer of them. It was like one nice little pocket.
True.
Yeah,
Yeah, smart call.
can check that objective off the box.
Of course, I said that, and then we just went right over one.
Just one
That's
though.
okay. We got 5 before.
It's been a while since we've seen them too.
Mm-hmm.
Oh, a tiny little thorny head.
Oh, so small.
What do we have here? Another snailfish. One
Nice.
off the right there.
Mm-hmm. Are they slimy? Is that why they're called snailfish?
That I don't know. Let's see if my book has something to say about it. It does not say anything about it.
They're just so darn cute. Love
They're
a
very
snailfish.
I've got another fish guide that is in another room that would probably have more information, but I would have to go get it, and it's also a 3-volume tome, so it's a little unwieldy.
Oh.
Why don't you come up a little?
There's a sea pen. Hopefully we'll see a few more. Can we pause and just zoom in on these sea pens real fast? I just want to see if they're different types than we were seeing earlier.
Yeah, no problem. Set it down here.
Those ones in the background? Or
Yep. Yeah, the ones in the background.
the brown puffs?
Yep, the brown puffs.
Okay.
I think those might actually be amphiptyllum, but I can't tell from this angle.
Yeah, they look quite bushy.
Yeah, I think
Send them there.
they're just laid over in the current. Yeah, so that is amphiptyllum. That's amphiptyllum grandiflorum. It's just laid over. So looking very low to the ground and just like the little brown puff.
Right on. What are the little white patches?
Bridge now.
I'm not sure
What
what
is
those
that?
are.
Tiny bacterial mats, maybe? Look branchy.
Add one more step, 100 meters. Thank you.
Okay. Should we move on? Moving on.
Sure.
Yeah.
Oh, there's another dead sponge.
Oh, it's a big one.
Yep.
Maybe there will be more in this upcoming slope feature.
Do you want me to reduce speed,
Fingers
Dan?
crossed.
See if I can get back out in front of it.
Sure.
It's pretty flat still. What is our bearing again? Was
290.
0290, thank you.
That pecan we saw is a species that's actually bioluminescent at night. It can
Oh, cool.
flash bright blue.
Cool.
So I'm thinking about holding position soon, and then we'll head up slope to AB. Unless you want me to zoom out here, you can.
No, that sounds good.
Yep, that sounds good. Just switching camera views here on my monitor.
Large crab coming into view.
Tanner crab.
Are those commercially fished?
They are in places.
Bridge nav. Hold position here. Thank you.
There's a nice
And
sponge. Going on another sponge.
That's cool.
Reorient to something like 020.
All right.
Looks
Can
like
zoom in there
That's
if
got
you want,
another C pin behind it.
Steve.
wow. What is that thing? Oh, it's a
sponge.
yeah. Just saw the
And
sea
a C
star.
pin behind it.
The sea star looked like its base, and it was really confusing me.
Bring your head around, Jake.
Yep.
Actually, looking at it, this is a sponge species that is on our target list if we see another one. That's just something to keep an eye out for.
You don't want to collect this one?
I'm trying to be good because we're supposed to wait till we see more than one.
It's pretty tough.
Do you suspect that they might be rare?
I don't actually have a good feeling for it. I know it's one of the species that Abby has on our list, but I don't know how rare it is.
Got it. If we suspect that we might not see one again, that's reason enough to collect it.
Okay. Well, if you are okay with it.
Yeah.
Okay, then we can probably go ahead and proceed.
So,
I like to think of myself as a reasonable person.
ship holding position, is
Ship's holding position, yep.
it? Right.
Resetting DVL. Looks like we're all good.
It is a beautiful specimen.
It really is.
Mm-hmm.
You want the whole
One thing
thing,
with some
or
of these is once you get them on the surface, they can be somewhat deflated. So you got to get the nice shot of them while they're underwater.
Can I just get this thing that it's on there?
If you don't think it's too big, that's a big chunk of dead heterocony sponge that it's growing on.
It's that whole piece.
That might be quite a big piece, and it will basically be like a giant rock.
Yeah.
Hey, fish.
Well,
Ooh. Ooh, so jello-like, that sponge. That's so cool.
You can see the oscula are starting to close as it's kind of jostled.
I can try and break the piece
yeah.
of it off, or
Oh, yeah, that would be fine.
oh,
Yeah, so the heterocony, the skeletons, like they're silicate and they fuse. They basically become rocks.
basically it feels like a rock.
Yep.
So how do you get this guy? You just grab
If
him?
you just grab him.
Alrighty.
the texture on the sponge change as it's pumping water out in response to being tapped.
Does he puff back up?
Yeah, well, if it's in water, it'll puff back up, and actually, yeah, if it stays a little squished, that's okay, as long as we can get some clean surfaces for spicules.
So I'm gonna have to smash it quite a bit to get it off there, it looks like.
Yeah, that's okay. Yep, and it'll puff up back up a bit in the box.
Yeah, copy. It could probably go in A, I guess. We have nothing in there, right?
Nope, there's nothing in any of the boxes.
Yeah, let's go for A. See if we can get these snaggle teeth into A. Come out. Looks like it.
Yeah.
Yep. All right, coming closed.
Taylor, and that was 063?
Correct, 063.
Gotcha.
Nice job.
We'll have a happy Abby in the morning. Thank you.
Our pleasure. Which way were we going again?
We're going to go 020, upslope.
020. Which way is Argus coming around?
Argus is to your, it's on your port side.
Is that a good way
Oh,
for
I
you
see.
to come, Jake?
I see what you mean. The wraps is what you're
Coming to
saying.
the right.
The wraps, yeah. 8 turns on the Argus only dive.
Well, when we went